In this video, it's the first of 4 videos on proteins and amino acids, and we're going to look primarily at the physical properties and the acid-base behavior of amino acids. Okay, so right here, we have an example of a protein or a polypeptide molecule, and what you'll notice here is that it's if you performed a classic amide hydrolysis on this, what you would find is that you cleave all of the bonds here, like the carbon-nitrogen bonds that are parts of amides, like right here, right here, right here, and those would convert to a carboxylic acid and an amine. So since each unit has both an amine and a carboxylic acid, the hydrolysis produces a series, in this case, of 4 different compounds that we're going to call alpha amino acids. So if we look at the standard nomenclature of a carboxylic acid, we call that the alpha position, and there's an amine group right there. So that's the immediate product of an amide hydrolysis, but a further reaction occurs here because you have a strong base, an amine, and a relatively strong acid, a carboxylic acid, and these are going to self-neutralize to give you this form here, which we're just going to call the zwitterionic form, or the dipolar form. Let me just write that out. So this is the and notice how my equilibrium arrows there go toward the zwitterion. The equilibrium favors the zwitterion over the compound with both an amine and a carboxylic acid. And that's just a acid-base neutralization reaction. So we're protein molecules are composed of amino acid units. A protein molecule can be quite large, composed of 500 or more amino acid units. I think insulin is considered a very simple protein, and that only has 51 amino acids, or only produces 51 amino acid units upon hydrolysis. It turns out that in nature, these 20 amino acids that you see in this table here account for more than 99% of the amino acids that you commonly find in proteins. I'm not going to go through this table, just use this table as a reference for the structures of the common amino acids. Notice that each of these amino acids, it has a proper name, then it has a 3-letter abbreviation, which is in organic chemistry class, that's typically what we stick with because our structures don't really get that complex. But if it gets really complex, then you go to a 1-letter abbreviation. I like the 3-letter abbreviations because they look like the actual names, but in some cases, when you go to the 1-letter abbreviations, they don't relate to the name at all. Consider phenylalanine is 3-letter abbreviations PHE, but its 1-letter abbreviation is F. So let's look at a few interesting things about these amino acids. So these 20 amino acids account for about 99% of the amino acids found in proteins. If you look at all of these structures here, all of them are chiral. They all have a chiral center. I'll just point that out here, or most of them do. So there's a chiral center there, there's a chiral center, there's a chiral center, but only one of these amino acids, glycine, does not have a chiral center. At the alpha position, you have 2 hydrogens. So that's unique. All of them have the L configuration if drawn from this perspective right here. And this is how we draw sugars. We draw amino acids the same way, with the carboxylic acid group at the top and the carbon chain continuing along the backbone, or on the vertical part of the Fisher projection. So when you write the amino acid from this perspective. The ones that occur in nature have the L configuration. When you assign the RS configuration, you find that all of them are S except for cysteine, this one right here, which and the only reason for that is because it has a sulfur pretty close in, and that causes the priority numbers to switch. In most of them, the nitrogen is priority 1, but the carboxylic acid is priority 2, but here the side chain becomes priority 2 instead of the carboxylic acid because of that sulfur. Okay, all of them are primary amines except for proline. Let's go back and look at there. So when we write it in the protonated form, they're all NH3 pluses, and you see that is a feature of all of them, right? But not proline. Proline only has an NH2 plus, and it has 2 alkyl groups at nitrogen. So it's unique in that it's secondary. It's a secondary amine. We call valine, leucine, isoleucine, phenylalanine, methionine, lysine, arginine, histidine, tryptophan, and threonine are what we call essential amino acids. Humans do not have any sort of biochemical process occurring in their bodies that make these amino acids, so they have to come from the dietary sources. And if you go to any health food store, you can buy any of these amino acids in their pure form. Okay, so if you're a vegan, you have to be aware that there's not a single plant that provides all of these, so you have to vary your protein sources among, beans and nuts and grains and things like that, so that you get all of them. You can't, whereas your average steak would provide all of them. Okay, all right. So anyway, let's look at some one of the most interesting physical phenomena about alpha amino acids is their acid-base behavior. All right, so when we write this Vitter-ionic form, it has a plus and a minus charge. They cancel each other out, so we're going to call this the neutral form. If we take a strong acid, we're going to protonate the carboxylic acid unit, and overall we have a +1 charge, so we're going to call this one the cationic form. And if we have a strong base, we're going to pull this proton off of nitrogen, and overall we have a single negative charge, and this is the anionic form. Okay? We call the transformation of the cationic form to the neutral form, the first pKa, or pKa number 1, and the second mole of base would be transforming the neutral form into the anionic form, and that's going to be pKa number 2. All right, so here's the Henderson-Hasselbalch equation. That's going to give us the basic relationship we need here. If the pH is below the pKa, we have predominantly the protonated form. If it's above the pKa, it's predominantly the deprotonated form. So the cationic form is the most abundant species if the pH is below pKa number 1. The anionic form is the most abundant species if the pH is above the pK number 2, and in between, the neutral form is the predominant species. Okay, this simple relationship here is really only good for the neutral amino acids, the amino acids that aren't carrying an extra acidic or basic functional group, and we're going to find that there's several that actually have additional carboxylic acid groups or additional amine groups. All right, a special value that is characteristic of all amino acids is pi value called the isoelectric point, and this is the pH where the concentration of the neutral form is at a maximum. If we have an amino acid that doesn't have any extra acidic or basic groups in it, the pi is about 6, okay? And it's very close to the average of pK number 1 and number 2. All right, if an extra acid or base group is present, here's the relationship. The stronger the extra acid, the lower the pI. And the stronger the extra base, the higher the pI. And let's look at a few titration curves here. This is the only titration curve in the organic chemistry class, probably. Okay, so a neutral amino acid, the pI is the average of pKa number 1 and pKa number 2. Okay, so right around 6 here. Okay, and this is where we've added exactly one equivalent of neutralizing base here. Okay, so we're going to start out in this titration with the cationic form and pure form. Okay, as we add more and more base, the solution, the pH of the solution gets higher. Okay, and when we've added one, pH here is equal to the pI, and it keeps getting higher and higher. I have this color code here because in this yellow area we have overwhelmingly the cationic form present. In this green area here, overwhelmingly what's present is the neutral species. At the high end of this, it's in equilibrium with the basic form. In the low end here, it's in equilibrium with the acidic form. Okay, and when the pH is really high, like in this purple or lilac region here, we have predominantly the anionic form. Okay, so this is alanine, very simple amino acid. The pKa1 is 2.34. This is more acidic than carboxylic acids. You might remember from the previous chapter on carboxylic acid derivatives. What gives here? Why is this more acidic? Well, because of this cationic group here, this is a really strong electron withdrawing group, so it's enhancing the acidity. Okay. Okay, so instead of being around 4, it's around a little lower, around 2. Okay. Now, the second deprotonation is the deprotonation of an ammonium salt, and this is right almost exactly the same as the pKa you remember of simple alkylamine, so it's very similar. Okay. All right, and halfway between these two pKa's, if we average them, we get the pI. Okay, here's an acidic amino acid, aspartic acid. Okay, it has 3 pKa values. All right, so pKa1, we're going to start with a cationic form here. The first, or the most acidic proton is this one right here, and it has a pKa of 2.1. The second most acidic proton is the other carboxylic acid proton, and when it goes away, we get this species, which is the anionic species. And we still have another acidic proton remaining, and this is, gives us the dianionic form. Okay, so when we're looking at the pI, we're looking at the maximum concentration of the neutral species. So we're looking at the transition, average of transitioning from here to here and transitioning from here to here. So this relationship holds that the pI is still the average of the first 2 pKa's. Okay, so here is the pI for the aspartic acid. It's right at 3, and it's the average of the first 2 pKa's. Okay, and what reason the amino acid is having such a much lower pI value is because the pKa2 is from another carboxylic acid. So that is making it much lower than the second pKa from a neutral amino acid like alanine, shown here, because the second pKa is the removal of a proton from the ammonium salt, which is not quite as acidic as the carboxylic acid. So hence we end up with the lower pI value. Okay, so some other groups that will have another acidic proton, most obviously is glutamic acid. Okay, so that has another carboxylic acid group. It has a pi slightly above here. This is because the two carboxylic acid groups are a little bit further apart. They don't talk to each other as strongly, so they're not quite as strong of an acid. So this pi is 3.2. Okay, tyrosine has a phenol group in it, and it's not really all that different in acidity from the ammonium group, but we do see a slight decrease in the pi value down to 5.7. Okay, the SH group here is actually more acidic than the amine group, so this is having a bigger effect in the amino acid cysteine, and its pi is down at 5. Okay, all right, so let's look at a basic amino acid. This is the most highest pi of them all, arginine. Okay, so when it's fully protonated, it's actually dicationic. Okay, removal of this first proton here gives us now the cationic form. And we don't get to the neutral form until we pull the second proton off. Okay, so here's the pi for the basic amino acid. Okay, and what we see here is that it's the second pKa and the third pKa that are transitioning from cationic to neutral. Okay, all right, so what we're looking at here is the result of adding two equivalents of base, and our pi value is the average now, not of pKa 1 and 2, but of pKa 2 and 3. So that's where we achieve neutrality sometime between the second and the third deprotonation. Okay, so when we average the pKa 2, 9, pKa 3, 12.5, we get right here at 10.8, and that is the pi for arginine. Okay, other basic amino acids are lysine, which has an extra amine group, and its pi is a little bit lower at 9.8, but way above the 6 that we see with the neutral acids. And histidine has this nitrogen here is basic in the imidazole ring, and this gives us a pi, again, quite a bit above 6 at 7.6. All right. We have 3 amino acid classifications. We kind of gone through them. Nonpolar, it means there's no acidic or basic group in the side chain. The pi is roughly 6. Here's 3 examples. Alanine, isoleucine. Okay, I put too many L's in isoleucine. Sorry about that. And methionine. All of them have PIs around 5.74. None of the side chains carry any really seriously acidic groups. Okay, we have acidic amino acids. These carry an extra acidic group. Okay, aspartic acid is the lowest pi of the 20. Glutamic is next, and cysteine with the thiol group is about 5.1. Basic amino acids, these are carrying an extra base group. The pi is way above 6. And the more basic the group, the higher the pi. Okay, so lysine's 9.7, histidine is 7.6, and this is 10.5. And if we look at the basic nitrogen, that's nitrogen that's sp2 hybridized. Okay, this is an amine. This is a type of group we haven't covered before. This is called a guanidine. And that's more basic than an amine. Okay, so the guanidine is more basic than the amine, which is more basic than the nitrogen of the imidazole. Okay, so we're going to end this video with a problem here. these are unnatural amino acids, but if we made them, we could figure out where they would rank in terms of their anticipated PI value just by knowing what we know about acid and base strength. In a way, this is a acid rank the relative acidity, rank the relative basicity, kind of conglomerated and asked at the same time. Okay, so we're being told here in the directions to place number 1 under the lowest PI acid. Okay, so let's look at what we can tell about these amino acids. All right, the first one's a neutral one. This one has a weakly basic group. The next one has a strongly basic group. The one in the lower left has a, let's say, a moderately acidic group. A moderately acidic group, but we know that fluorine is actually going to make that a stronger acid, so we're going to rank this fluorinated carboxylic acid ahead of the non-fluorinated carboxylic acid. A phenol really isn't all that acidic, but it does have some degree of acidity. And this last one is a sulfonic acid. This is an organic analog of sulfuric acid. So this is actually a very strong acid. So we rank these. We're going to rank the one with the sulfonic acid is number 1. Okay, we're going to rank the one that's got the fluorine next to the carboxylic acid as 2. We're going to rank the regular carboxylic acid, which is actually, that's not an unnatural amino acid. That's just glutamic acid there, isn't it? Yes, so number 3. All right, this is going to be number 4, this one with the phenol group. Okay, and next would be the neutral, and of the 2 basics, the weaker base is going to, or the more weakly basic one is going to have the lower PI value, so that's going to be number 6, and then number 7. Okay, so we're going to end this video here, and in the next one we're going to look at how to prepare amino acids.